Welcome to the last unit of Chem 101 for the spring 2020 coronavirus interrupted semester. So when we were trying to decide how we wanted to finish out the course, we were really trying to figure out what we think is important for you to know and understand going forward as people in healthcare, and the role that chemistry actually plays in studying and developing treatments for diseases, novel diseases such as the COVID-19 that we are sort of all experiencing in our own ways this year. So instead of going into the last unit, which we don't think is as important for this and is not required in the course description, we decided that we would give you a little bit of information about viruses in general, the chemistry of a virus, the chemistry of infection, the chemistry of treatment, and how this relates to some of the things you might have learned in this class. So we are going to be presenting you two different kind of case studies. The first one is contained in this PowerPoint, and I'll narrate through for you. The second one will be a case that you will read, and then there'll be a short exam, which will count as your last exam for the class, where you can use the information that you've learned on these two presentations. For this exam, feel free to reference back to the material because it is a little outside of what we've done. So I'm not expecting that would be completely closed book, but you should be able to answer all the questions just based on the material that we're presenting to you in this PowerPoint presentation and in the other case study. So there is a National Center for Case Study Teaching and Science, it's the NCCST, that provides all these different case studies to science teachers if you subscribe to the site, which I do. And in the last 6 weeks or so, almost every case that I've been receiving has something to do with coronavirus. And a lot of them have to do with anatomy and physiology. So you may find when you take anatomy that some of those cases will be used. So I'm going to present you one that I got at the very beginning of, right before actually a pandemic was issued. So this was before COVID-19 had become a global pandemic. And I actually presented this to biology in person right before we took our learning onto the remote platform. So this presentation is entitled Outbreak. I've edited it down so it doesn't contain all the slides that it had when I first got it. And it starts with a patient case, a diagnostic pathway, and then we're going to talk a little bit about how viruses infect cells and the chemistry behind that. You could also talk about the chemistry of the testing. I'm not going to go into details about how all the different testing works. I just want you to kind of get sort of an overview of how chemistry is important in the development of all of this and help you to think of chemistry as really being the central science that guides really all of biology as well as all of chemistry. So the title of the presentation is Outbreak, and let's go ahead and get started. So you are a nurse in a clinic, and a patient presents with the following vital statistics. So vital statistics are things like age, sex, height and weight, blood pressure, temperature, sort of easily measurable diagnostics. And as a nurse, and even as another type of healthcare professional, you might be taking these kinds of vital statistics. So this is a male, age 10. He's height 4 feet 5 inches tall, weighs 70 pounds, normal blood pressure of 120/80, but he's running a fever. So normal body temperature is about 98.6 degrees Fahrenheit. His temperature is 103 degrees Fahrenheit. This is America. We measure temperature in Fahrenheit in the doctor's office, but that would be a fever. He has the following symptoms. So he has the fever that you've just diagnosed with your taking his temperature in the office. He's complaining of cough, shortness of breath, which means that it's hard to get a deep breath. So people feel like it's like their chest is tight. And it's reported that this has lasted for about 1 to 2 weeks. So these are symptoms that could be lots of things. Fever, cough, and duration of 1 or 2 weeks definitely could be flu. Shortness of breath, not so much, but people do get pneumonia from the flu, so it could still be flu related. It's probably not just a simple cold because usually colds don't last that long and usually don't result in fever. So now, what do we ask the patient about? So now that we know more about COVID-19, we certainly would think about COVID-19, but this is kind of a novel presentation. So at the beginning of this pandemic, before it was a pandemic, when the first cases in the United States were being isolated, there was discussion of, did this patient travel? Did this patient have a family history of anything? Does this patient have a chronic underlying illness or something that's congenital that they've had from birth, something like a lung disorder or something like that? So when we think about what we should ask patients about, we should definitely ask about family history. We should ask if they've been anywhere outside of the country or even outside of their local where they live. And we should ask if they have any chronic conditions. So all of these questions would be things that would be relevant. So you go ahead and you ask the patient all of these questions. And the patient reports that there's a family history of hypertension, which is high blood pressure, but the patient himself does not have hypertension. There's no history of cancer, diabetes in the family. They don't mention any other chronic conditions, so presumably none of those. No one has traveled. They live in the US, and he has no other chronic kinds of conditions. So then the question is, and we've heard a lot about testing in the last month, how do you test? What do you test? So we're going to talk a little bit about some of medical testing, some of which relies on fluids from the patient, some of which is things like x-rays, etc. So these are the tests that could potentially be ordered to help diagnose this patient. A sputum test is just taking basically saliva and analyzing it for the presence of bacteria or fungus. So saliva you can't test really directly for virus. The flu test, which is a nasal swab, actually gets some tissue. So you're looking for the presence of infected cells with the virus, because that's where the virus would be. So this is called the RIDT, the Rapid Influenza Diagnostic Test, and that's a nasal swab. And then more expensive and tests probably that would come further down the road would be some things like chest x-rays or MRIs, magnetic resonance imaging, both of which chest x-ray, you're exposing someone to radiation, so you probably don't want to order that if you don't need it. MRI is very expensive, takes a long time. Put a 10-year-old in an MRI, they have to lie still. Probably not the first test you're going to order. So you go ahead and you order the sputum test and the RIDT test. Both of these are fast, and they both come back negative. So the sputum test says there's no bacteria or fungus detected, which means it's probably not a bacterial or fungal disease. The RIDT test is testing specifically for influenza virus, which is the virus that causes the flu, and that came back negative. So now you have to ask yourself, well, what's causing this disease? So when they say, what type of disease is this? What they're really asking is, what's the causative agent? Is it bacteria? Is it virus? Is it fungus? Because those are really the three things that cause disease like this. Based on the sputum test, it shouldn't be bacterial or fungal. Based on the nasal swab test, that only tests for one kind of virus. So it's likely that it's a virus, but not the flu virus. So what is a virus? So for those of you that had biology, we talked a little bit about viruses in biology. For those of you that have not had biology, I want to give you just a snapshot of the virus cycle. So in this slide, it says life cycle of viruses. It really shouldn't say life because viruses are not alive. So we'll just call it the cycle of a virus. So a virus is a piece of some kind of genetic material, DNA or RNA. And we're not going to go into the differences, but there are different viruses that have different types of genetic material. But the first thing a virus has to do is to infect a cell. And it has to get into the cell to do that. So there's some way that the virus is going to attach to the cell and get into the cell in some way. Different viruses do this in different ways. Some viruses just inject their genetic material into the cell and the rest of the virus doesn't get in. Some viruses get in all the way. So we're not going to talk specifically about this happens. But what happens in both cases, in however the virus infects the cell, is the virus has to somehow attach to the cell. And that is going to be through the laws of chemistry. So the laws of chemistry are going to dictate how these molecules that are on the virus interact with molecules that are on the cell. And there has to be some kind of an attachment. And we're going to talk more later in this presentation about how that attachment works. So once that happens, the virus then is reproduced inside the cell, but only because the cell reproduces the virus. A virus cannot make more of itself. And if you remember from biology, one of the criteria that we establish for deciding if something is alive or not is whether or not it can reproduce itself. If it can reproduce itself, then we call it, that's one of the properties of life. Viruses can't. They rely on a host cell. So the only way a virus can be reproduced is if the host cell makes more of it. So viruses sitting on a petri dish will not make more of themselves because they don't have the necessary machinery. So the virus gets made, the cell makes not only more of the virus genome, it starts to make all the virus proteins, which are another kind of chemical substance, another molecule, and any other types of molecules that are required to package that virus back into its own little pretty package. And later we're going to talk more specifically about coronaviruses. After that, the cell often ruptures and dies and then releases all this new virus into the plasma of the patient, which can then go and infect other cells. And that's how a virus kind of takes over a body. So this is just a general overview of what they call the life cycle of a virus, but it's really just the cycle of a virus. So how do we test for the presence of a virus? So we've been hearing a lot about testing. And these are what we call serology tests. So we're testing serum. What you're testing for is actually two different things. So as you fight an infection, the way that your body fights an infection is through producing another special kind of a chemical substance, a special molecule that are your antibodies. And these are encoded in your genes, of course, but they're special molecules. And antibodies against a particular virus have chemical properties that allow it to interact with the virus and neutralize it. So again, all governed by the laws of chemistry, you have this interaction between an antibody and a virus. Okay. So there's a test called an ELISA. It's an old test. It's been around for years, but you need a specific antibody against a virus in order to be able to detect the virus. So if you have an idea of what kind of virus it is, or you can test with a whole bunch of different antibodies, it'll help you to establish what the virus is. And antibodies for this type of testing are often commercially available. Many companies specialize in production of antibodies so that you can just run these assays pretty easily. It doesn't take very long, okay? But it's somewhat nonspecific because sometimes you'll have an antibody to a family of viruses. So it'll tell you you're in this family, but it won't tell you which specific virus it is. So that's the first test. So if that's positive, then you want to get more and more and more specific. So the second test actually tests for infection. The first test is testing for the presence of an antibody, but that could be because you were infected a long time ago and you still have antibodies around. The second test is going to test for virus infection directly in a cell. And this is called an immunofluorescence assay. You need some tissue for this. And it's a little bit more, it's quite a bit more specific, and it detects active infection. So the first one detects antibody, which could be active or prior infection. The second test tests for active infection, so it's looking for viruses in cells. If both of the first two tests are positive, usually that means the patient is considered positive for that infection. If the second test is not 100% sure, then you can move on to the third test, which is much more specific, expensive, and time-consuming. And I don't know very much about that test, but that would give you the third. So if the second test is indeterminate or not sure if it's positive or negative, then you can move on and do the third test. So given this information, what tests would you now run? Okay. So initially, you're probably going to run a serology test. Okay. You're probably not going to run a biopsy or a spinal tap because there's really no reason to run a spinal tap at this point. There's no evidence that there's neurological involvement. And a biopsy is pretty invasive. You have to go get tissue. We didn't talk about molecular tests. And molecular tests are really important because you're testing actually for the presence of the virus molecule. And specifically, you're testing for the presence of the virus genetic material. But you can only do a molecular test if you know something about which virus it might be. So after the serology test confirms, yes, I think this is a coronavirus, then you can move on to molecular tests, which can give you much more information of what specific virus you're looking for. So the serology tests in number 2 were all positive. They tested for the presence of 2 different proteins, which are molecules, chemical substances, that are on the virus particle. So when they say nucleocapsid, nucleo is for nucleic acid, that's one type of molecule. Capsid is the capsule. So viruses are like little capsules. They protect their RNA or DNA. And then spike is a protein we're going to talk about later. Spike is a very important protein for a coronavirus because that's how it helps it get in the cell. So those were both positive. So then they did a molecular test where they actually looked at what genes were in this virus. And the molecular test indicated that there were 2 coronavirus-specific genes that could be detected. The test is called PCR. It's called polymerase chain reaction. It's a test that's been around forever. It's very, very sensitive. It can be done on all different kinds of cells. Sometimes you only need one cell to get a positive result. It's that specific. So after all of these tests were done, all the evidence should lead you to a diagnosis. So what's the diagnosis? So here are the choices. Influenza, well, we know it's not that because the rapid influenza diagnostic test was negative. Coronavirus, well, we've got 3 positive serology tests and a positive molecular test for coronavirus. Common cold, which is a rhinovirus, didn't test positive for that. Lyme disease, that never even came up. Lyme disease, it's not Lyme disease. Okay. So the diagnosis for this patient in this case is coronavirus. Okay. So what do we mean when we say coronavirus, and how is COVID-19 related to this coronavirus in general? So there are lots of coronaviruses. Chances are you've been infected by a coronavirus at some point in your life because typically they aren't very dangerous. They give you a mild respiratory infection, much like a common cold or a mild flu. You might not even go to the doctor for it because you're just thinking you have a cold. Okay. So the symptoms are so similar to what we see with a common cold that most people get this infection at least once in their life. Coronavirus is not one virus. So in the world of virology, which is a really complex, crazy, and super interesting area of science to study, I think it's almost even more complicated than studying living things. There are enormous numbers of different types of viruses. And in each what we call family of viruses, there are enormous numbers of different viruses in there. So coronavirus gets its name because of what it is. It is a virus that has RNA as its genome, RNA molecule. It has a specific structure outside, and there are proteins that stick out of the side walls of this virus that give it the corona name because they look like crowns. So it can cause diseases ranging from common cold type symptoms to very severe diseases. And in the past 20 or so years, there have been several more severe coronaviruses, none of which reached pandemic status until this last one. So back in the early 2000s, there was a disease that came out of China. It was the South Asian Respiratory Syndrome, or SARS. We're now calling that SARS-1 or SARS-1. There was another one that came out of the Middle East in the 2012-ish time called Middle East Respiratory Syndrome, or MERS, and both of these never reached pandemic status. I looked it up the other night, and I think there were 8 cases of SARS-1 in the United States and fewer than that of MERS. MERS was very deadly, but it didn't really spread very much from person to person. Okay. So most coronaviruses have an animal origin. They're called zoonotic, zoonotic diseases, because they come from an animal source, and something changes in the virus that allows it to be transmitted to people. In the case of COVID-19, we've had a change that allows it to be transmitted between people. So typical symptoms of coronaviruses: fever, cough, shortness of breath, sometimes pneumonia. Very infrequently, you have some kind of intestinal, sometimes asymptomatic, because we've heard of this. And this is for MERS, but it's pretty similar to what we're experiencing with COVID-19, except that COVID-19 is spreading between people. So diagnosing, same tests we just talked about, molecular tests, serology tests, etc. And then this is just a slide kind of again going through the diagnostic process. So who is most commonly affected by COVID-19? The people that are most commonly affected are older people, people with weakened immune systems, people who have chronic diseases such as renal disease, which is kidney disease, cancers, chronic lung problems, diabetes, hypertension's been in there, heart disease. In this particular coronavirus, young children don't seem to be more affected. As a matter of fact, young people seem to be a little more resistant for whatever reason. So now let's move on to how this has something to do with what you've learned this semester. So you have a little bit of virology. So you might be thinking to yourself, what does this have to do with my chemistry class? And the important thing to remember is it has everything to do with chemistry. We're going to talk a little bit about viral getting into cells. We're going to talk a little bit about therapies, the testing. It's all because of chemistry. So remember that all matter is made of atoms. Atoms combine to form molecules. In biology, we have big molecules, but they're still chemicals. Everything in biology, everything that happens in your cells can only happen because of the laws of chemistry. So even though we look at cells and we think, oh yeah, the cell's doing all this biology, but then I go to chemistry and they have nothing to do with each other, they have everything to do with each other. And everything you learn about when you go to anatomy, when you go into nursing, when you start to talk about gases and patients and respiration and everything has everything to do with chemistry. So for things to happen in biology, these very large biological molecules must interact in specific ways, dictated by those laws of chemistry that we have been learning about all semester. Laws derived from that periodic table, that organization of the periodic table. So let's talk a little bit about the chemistry of COVID-19. So the first thing we're going to talk about is how does this virus actually infect a cell? Okay. So on this virus, remember we have this lipid coating on the outside, this layer of fat that surrounds the inner part of the virus. And sticking up, it's almost like a cell membrane, sticking up through this layer of fat are several different molecules. And the molecule we're going to talk about right now is a molecule that is called spike. S. We could just call it S, but it's called spike. And spike is a very specific kind of molecule. It's called a glycoprotein. What's a glycoprotein? So let's break the word down. Glyco, think glycolysis, sugars. Glyco means sugar. So this is a sugar protein. So this is a molecule that has a protein and a sugar attached together. And those types of molecules are often involved in things that happen on the surface, because they're really good for immunology, they're really good for specifying interactions. And this particular protein, this glycoprotein, has a very specific chemical interaction with a binding site on cells that is the receptor for an enzyme called ACE2, which is an enzyme involved in regulation of blood pressure. And you may have heard of ACE2. You may not have. You're certainly going to hear about it when you talk about regulation in anatomy class. So ACE2 is on the surface of cells, and it's there for a normal reason in our bodies. It binds to another protein, but it also binds to this spike protein, the spike glycoprotein. And this interaction is controlled by all the laws of chemistry. So on the next slide, this is a very lovely modeling of the interaction between the virus, which is on the top of the slide, and the spike protein, which is this big red protein that is sticking out of the surface of the virus. So this layer up here that looks like little matchsticks that have purple heads and little wood, this is that phospholipid bilayer. This is that fat layer on the virus. And then up here further beyond that we can't see on the slide is where the inside of the virus would be with its RNA molecules and other molecules that it needs to get infection going once it gets in the cell. So don't worry about all this. You don't need to worry about subunits and whatever. But this interaction, dictated by the laws of chemistry, allows this virus to bind, and then it will be internalized into the cell. So this virus gets into the cell, they believe, by a process called endocytosis, which we talked about in bio. I'm not going to go through it again, but it's a way that the whole particle can be brought in. So after the virus gets into the cell, this kind of shows you what's going on in the cell. The cell makes more viruses. Now remember, viruses are not alive. They can't make more of themselves by themselves. They have to get into a cell so that they can be what we call replicated or reproduced. Everything that happens in the cell to this virus is dictated by the laws of chemistry. I'm not going to review all the biological processes that occur. You're not going to be asked about all the different biological processes that occur. All I'm going to tell you is that once it gets in, you see all this activity, more and more virus gets produced, and then the cell is going to spit out all those viruses that have been made, and those viruses can then go and infect other cells. Multiple viruses can get into a cell at the same time. They're all being copied over and over again, so you can see how this infection can just ramp up and really spread. So how do you treat for viruses? Okay. Treatment is generally right now supportive and based on the patient's clinical condition. And you may have been hearing a lot in the news, if you listen to any of the press briefings, certain types of medications have been tried. There are several different types of pharmaceutical compounds that you can use for infectious agents depending on what the infection is caused by. Antibiotics are pharmaceuticals, chemicals that are developed by chemists that are used to seek out, treat, and destroy diseases that are caused by bacteria. Okay, so bacteria, remember, are prokaryotic organisms. I'm not going to go into a whole difference, but because they're not the same kind of organism as people, you can develop drugs that specifically shut down the ability of a bacteria to reproduce without affecting the cell that it's in, because the mechanisms are different. Remember, viruses don't do that. They don't have their own reproduction mechanism. They use the cells. So molecules that are effective on bacteria, antibiotics, are not effective on viruses because they don't shut down that process in a virus. Okay. There are drugs called antivirals. Antivirals are really complex because antivirals have to seek out a part of the virus cycle to block. But remember, every family of viruses has a completely different mechanism of how it infects a cell, how it's reproduced by a cell. So how to target which antiviral is going to work is really much more complicated than an antibacterial, for example. So I used to always think it was better if my kids got strep throat because I knew that I could give them an antibiotic and it would cure it. Whereas if they got, a bad viral disease, you just oftentimes have to wait it out. Things like chickenpox, which nobody gets anymore because of the vaccines. So in the absence of a vaccine for a viral disease, a lot of treatment is really just palliative. There's a lot of people working now on developing specific antivirals for COVID-19. So an antiviral has to disrupt the viral cycle somewhere. So antivirals that have been developed for things like HIV, which is a completely different family of viruses than COVID-19, aren't going to work on COVID-19. The mechanisms are different. So the molecule has to be developed to interfere specifically with that virus. So that is the end of our presentation. The goal of this presentation was to make you appreciate how important chemistry is in understanding biology, specifically in understanding infection, infectious disease, the development of vaccine, the development of treatment for viral infections. So when you're done with this, go and read the case study, and when you feel ready, go and take the final test for the semester.